not actually special types of microscope. It is just a variation in the compound microscope, which we have already studied. So first is the dark field microscope. When do we use the dark field microscope? When the specimens and their components, they have the refractive index close to the medium in which they are suspended, and thus they are very difficult to see by the bright light microscope, in which we see the field of view is bright, and on which we see the colored specimen, which is done either by the staining of the specimen, and thus which we perceive the image of that slide. So in dark field, the background is dark and the sample in the specimen is either unstained or it is white. And to obtain this, we use a dark field condenser which contains the opaque disc, which blocks the light coming from the below. This light source is blocked off, which causing the light to scatter when it hits the specimen, and only the reflected light enters the objective. So just to recapitulate, the bright field microscope, here we have the light source. The light passes from this to the condenser lens. The function of the condenser lens is to condense the light falling on the specimen. The light from the specimen is then reflected to the objective lens, which magnifies that image, and that magnified image then passes to the ocular lens, and which further magnifies this image, and we perceive in the eyepiece. But in the dark field, what do we have? We have an opaque disc, a black disc. Now, when the light is falling on this disc, the light in the central part is blocked, and only the light coming from the sides, they fall on the condenser lens, and then we see this light is then falls on the specimen or the stage. Now, think if there is no specimen in this stage, then only we will see, we won't be able to see anything, and it will appear only a dark. But if there is a specimen or a section or then the light gets reflected, and then only the light which is being reflected by the specimen is captured by the objective lens, and then this is perceived by the ocular lens and eyes. So this is the optical disc or the dark plate, which is kept on below the condenser to block the light coming in the central part, and no direct rays enters the object, only the scattered rays from the edges of the structure within the specimen, they form the image. Like we can think, if it is a dark room and a light, you are sitting in that room and a light falls on you, then the room is dark and you are being illuminated. This is same. This is just simple compound bright field microscope with a dark patch by which we block the central light of the microscope. So when there is no sample, field of view is dark as there is no diffracted light. But when there is a sample, some lights are diffracted which are perceived by the eyepiece. This gives a high contrast to visualize the shape and the structure of the specimen, which is unstained or light colored. This is the same. So what is the advantage of this dark field microscope over the bright field microscope? It is simple and is ideal for objects which are unstained or transparent. And basically it is used to study the external features, and contrast is therefore better than the bright field when the specimen or the slide is unstained. Second special type of microscope is phase contrast microscope. In this also, the specimen here we take is unstained or light stained. But what do we see? Overall structure and the shape of the specimen, it is provided with crisp details without staining the specimen. And what do we add? We add one face plate. Apart from the dark plate which has been added on below the condenser, we add a face plate. This face plate is added between an objective and the eyepiece. This face plate is like a circular thing which has two portions. One portion is coated with light retarding material and the other portion is coated with light unretarded material. So what is this phase? Phase contrast microscopy. What do you mean by phase? Phase is a relationship between two signals with each other. This phase relationship is important because it enhances the contrast of the transparent and the colorless object by influencing the optical path. So the optics is, here we have the light source. Light then first passes to the dark plate or the annular diaphragm, and only the light through this central hollow portion, it falls on the condenser. Condenser condenses the light onto the specimen, and when the light passes through the specimen, there are two types of rays which are generated. One is the retarded rays, which is shown in the dotted line, and the other is the unretarded rays which pass through the object, which does not pass through the objective, and then these form an image. This retarded, suppose if there is a cell and in the cell there is a nucleus. So when the light is in a wave form, when the light passes through this nucleus, it gets retarded. That is, the peak of the light rays gets slowed down. But when there is no organelle in the specimen, then the light moves through it directly. So, but our eyes cannot see this change in the wavelength of the specimen of this light. To overcome that, we add another face plate between the objective and the eyepiece. So what does this face plate does? This face plate, it will reduce the wavelength of the retarded rays by λ/4 and increase the wavelength of the unretarded rays by λ/4, thus causing a change in variation of λ/2. This change in variation of this wavelength is perceived as a change in the intensity, and thus we are able to see the specimen. So phase contrast microscope, it works on the principle of interference. We have these light rays in the form of waves. So when there are two waves which have the same peaks and same troughs, when they combine each other, we get a wave which is more brighter, and that is being perceived by the eyes. But when there is destructive interference, that is when the wave, the peak of one wave is opposite to that of the other waves. When we add these two, we get a destructive interference and we are not able to see the specimen, and that becomes a darker shade in the intensity and of the specimen. This is called as, the specimen is out of phase, and the brighter part is called as, the specimen is in phase. So light gets retarded due to increase of refractive index of the specimen because of the organelle that is present in the specimen, and which causes the decrease in the wavelength. After passing through this objective, the light ray falls on the face plate, where wavelength of these retarded light is increased and wavelength of the unretarded light is decreased, and causing a constructive interference with high intensity of light, which is being perceived as in the specimen. This is the same. How does this face plate looks like? We have two types of face plate. One is being coated, which retards the light, that is, it allows the light to go through it slowly, and the other is which does not allow the light, which allows the light to go through it more in a faster way. The application of phase contrast microscopy is to get a high contrast image of the transparent specimen. like the living cells in culture, to see the microorganism, or thin tissue slides, or a subcellular particle like a mitotic chromosome or a motility of a mitochondria, that can all be seen by the phase contrast microscope. Third special type of compound microscope is the polarizing microscope. Before going on to the polarizing microscope, what is a polarized light? Can anyone tell what is a polarized light? What is an unpolarized light which we see? The light that light is scattered in all the directions, that is called as unpolarized light. Polarized light, when the light that is moving only in one direction, that is a polarized light. So normal light that vibrates in many planes or direction, while the polarized light, it vibrates only in one direction with the application of a polarizer. Second question is, what is birefringence? We say certain substances are birefringent. What does it mean? It means that that substance has different type of refractive indices. There are different areas where the refractive indices are different in that substance, and that causes a double diffraction of an anisotropic substance. This means that the incident light which falls on this birefringent material, it splits into two rays. One is called as a slow ray and the other is called as a fast ray. So this polarized microscope uses the polarized light to evaluate the composition, and it overall gives a 3D structure of this anisotropic specimen. So in addition to the standard microscope optics, we have a polarizer which is mounted below the condenser, and the other is the analyzer which is mounted above the objective of the microscope. And the specimen that is illuminated with a plain polarized light, and the rotation of this polarizer and analyzer, it can be analyzed, and which we get a colored 3D image. So this polarizing microscope, it works also on the principle of interference, but we get a 3D image of the anisotropic specimen and is colored. So this is a schematic diagram of this polarizer. Here we have the light source, and then we have the polarizer. This polarizer limits the vibration of light in only one direction. And then we have a graduated circular ring or stage which facilitates the orientation of the specimen, and thus we can get a colored image in varying degrees. And then we have a compensator or analyzer which is placed between the objective, and it enhances the optical part difference in the specimen. And sometimes we have this Burton lens also, which allows the accurate adjustment of the illuminating aperture of the diaphragm and see the interference figure. So we have the light source which will give an unpolarized light. Then with the help of the polarizer, we get the polarized light, that is moving only in one direction. When this polarized light, it strikes the birefringent specimen, two types of rays are formed. One is the ordinary ray and one is the extraordinary ray. Now with the help of the analyzer, we try to rotate it and get a recombinant light rays after the interference, giving a colored image of the birefringent material. Polarizer is fitted below the condenser and analyzer is placed above the objective. As I have already told, there is a compensator which might be inserted between them. Now when the plane of polarizer and analyzer, they are perpendicular to each other. No light passes through the ocular lens and we get a dark image. When the polarizer and analyzer, they are at 45 degrees to each other, we get the maximum intensity of the light and the color of the image changes depending upon the orientation of the specimen. So here we have the polarizer. The light passes through the polarizer, light moving in one direction, which falls on the condenser. It condenses the light on the object plane. Then the light passes through the objective, giving the phase retardation of one way with respect to another, giving an elliptically polarized light, which is transmitted by the polarizer. Fourth special type of microscope is stereo microscope, a slightly modified version of which we have studied in the operative microscope. Here we use the dissecting microscope. This has a low magnification power. Earlier, the bright field microscope, the dark field microscope, phase contrast microscope, polarizer microscope, they have a high magnification power. But the dissecting microscope, it has a very low magnification power. And basically, it has two eyepieces. This is the stereo head, and it has two light sources. One is top lighting source, and the other is the bottom lighting source. This is different from that of a compound bright field microscope, in which we have only a single light source from the bottom. Here we have two light sources, and this is the objective, but the power of this objective is very low, so that overall total magnification is very low. Therefore, it is primarily used for dissecting of specimen and analyzing them. It uses the light reflected from the surface in comparison to the transmitted light which we use in the bright field or the dark field microscope. And use of this reflected light from the object, it allows the examination of the specimen that are too thick or opaque, that can be used in the bright field microscope. Moreover, the distance between this objective and the stage is larger, and therefore we can also see the solid stone, marble, or it can be used in the forensic applications also. So the applications are to study the topography of a solid substance. In medical field, we use it for dissection or microsurgical procedures, or to inspection of the fractures, and it can be used in forensic engineering as well. What is the advantage of this stereo microscope or the dissecting microscope? It uses two light pathways. We can think it as composed of a two monocular compound microscope. So it uses two light pathways, which offers a great magnification for the visualization of the image. It is easily portable and is ready to use, and we can view the specimen as a whole. The last is the inverted microscope. As the name says, inversion. Objective lens here, it is placed below the stage and the light falls on the specimen from the above. Now, where can this technique be used? This technique is used for live cell imaging, where cells are observed through the bottom of a cell culture plate. So here is the light source that is above. Condenser is here below the light source. This is the stage, and the objective are placed below this stage. The objective below the stage, it collects the light from the condenser and magnifies the image, which is then sent to the eyepiece as in the bright field microscope. The uses are to see the fungal cultures which are in a petri dish or a culture bottle, to observe the nematodes, and it may be used for the visualization of Mycobacterium tuberculosis in the technique of MODES, which is a drug susceptibility assay which they use, and to observe the living cells present at the bottom of the tissue culture plate or flask. Any questions?